All right, well, it is 10:30 and we like to start things promptly when it is time. I appreciate you all joining us today. I am Jennifer Mitchell. I am a public communications coordinator and have been organizing these webinars and appreciate you joining us today. Uh, Please know that you will be on mute throughout the presentation. We do hope that you do ask questions though. This is going to be a very informative presentation today about where we are going, or where are you going, Indian River Lagoon, what recent conditions might tell us about the future. Today, Dr. Chuck Jacoby is going to give us an overview of what's going on in the Indian River Lagoon and answer those questions that we have after his presentation. He is a supervising scientist, environmental scientist at the district, and focuses on the estuary. So I'll let you take it away.
Thank you. Good morning, and yeah, we'll get into it. Basically, I'm going to kind of go over some of the things we've seen recently in the lagoon, what's going on in terms of a response, and some things that we learned as we watched this go through. Hang on, there we go. So first off, probably should understand the system that we're looking at. Oops. So the Indian River Lagoon is a very valuable component of Florida's natural resources. A recent estimate puts its annual economic value at $7.6 billion. And that was just kind of looking at the 5 sectors that you see listed on this graphic here, with defense and aerospace and recreation and visitor related expenditures being among the largest. The system, it's actually a system. It's called the Indian River Lagoon typically, but it's actually 3 receiving water bodies, the Mosquito Lagoon, the Banana River Lagoon, and the Indian River Lagoon. All total, the system spans about 156 miles of the east coast of Florida. It's a shallow system. The average depth is about 3 feet. Although river is in the name of parts of the system, that really isn't a river. It's an estuary. It's a barrier island estuary. So an estuary is where the fresh and the salt water mix first. And this one is formed by a barrier island that formed offshore. So the system itself is not driven with a flow like the St. Johns River. It's driven mostly by winds and tides, and mostly winds. This means that the system is segmented, so parts of the system don't turn over, the water doesn't turn over for a period of months. In some places it's weeks, but in some places it's quite long, up to around a year in the more remote corners, as it were. All these things make it a sensitive system. It's vulnerable to having things such as nutrients, which is what we'll focus on today, or contaminants enter into the system because they tend to stay there. But it's also these same characteristics that make it a very diverse system. And its length also means it's got diverse politics. So the challenges in the southern end are a little different from the challenges in the northern end. And today we're going to focus in kind of on the northern part of the lagoon, which is part of the St. Johns River Water Management District. And it's not just the district that pulled together the information you'll see. It's a plethora of partners, as it were. This is definitely a collaborative effort to manage this system. So get into what have we seen recently, and recently now for this particular talk will be from 2011 on. So in late 2010 and through 2011, there were 2 blooms of phytoplankton or microalgae, so single-celled photosynthetic organisms, things that take light and convert it into biomass, biological material. There was a super bloom in the north, and the reason that we called it a super bloom because it exceeded the historical records, both in terms of intensity, how much algae was in the water, which we typically look at by looking at a pigment. That's part of their ability to capture light and use it, the photosynthetic pigment, chlorophyll. And the concentrations of chlorophyll were about 3 times what we had recorded in the past, or 2 times, sorry. And then the duration was about 3 to 4 times longer than blooms we recorded in the past. About the same time, there was a bloom further south that you can see in the satellite images as the sort of, if you go to October 1, you can see a kind of a green and yellow section. There was a bloom down in that was lower in terms of intensity, less chlorophyll, but it lasted longer. So the red colors on these satellite images are higher concentrations of chlorophyll. So you can see that it got pretty intense over a fairly large area, and it lasted a fairly long time. Following that, in 2012, we had our first brown tide. That's Umbra lagunensis is the organism that causes the brown tide. This had appeared in Laguna Madre in Texas earlier in the 80s and hadn't been a dominant in the lagoon. When people went back and looked at some of the samples that had been archived, they could find Umbra, but it had never been dominant. But in 2012, it was, particularly up in the northern part of the lagoon system. 2013, there's a series of different kinds of blooms. You can see here some brown tide, Umbra up in the upper left there, and some dinoflagellates and so forth in different other places. 2014 and 15 were a little better in terms of blooms. There weren't as widespread blooms or as many blooms. But 2016, we had another brown tide, which was very intense in Banana River Lagoon. Again, these are satellite images with the chlorophyll modeled from the light that reflects back to the satellite. And then that's verified or ground truth by using our field data. So the hotter colors are more chlorophyll, and you can see the scales there. 2016, this bloom that was in Banana River Lagoon actually led to a fish kill, a large-scale fish kill. But we saw a similar kind of blooming in 2018. It was a bit more mixed, but it reached the same sort of intensity of chlorophyll, and that was a concern. We fortunately did not have a fish kill from that bloom, a large fish kill. But we're also seeing the same thing now. So the right-hand satellite image is from the 27th of October this year. And again, you can see that Banana River Lagoon is pretty hot. The very tip of Mosquito Lagoon that you see at the top of that is also similarly got a lot of phytoplankton in it, and then northern Indian River Lagoon. And the white areas are the clouds. Satellite images can be challenging if the weather gets cloudy. It's hard to interpret them. So we've seen blooms over this span of time that kind of reach the same sort of extents again, you know, the aerial extent, and also the intensity, how much chlorophyll there is in the water. We did see some potential and actual realized flow-on effects of these blooms. So in 2012 and 13, there was an unusual mortality event for manatees. The cause has still not been determined fully. Some people try to link it to these blooms, potentially a change in the availability of what the manatees eat, because the shading of the phytoplankton caused less algae and so forth and seagrasses for them to eat. 2013, there was a dolphin unusual mortality event, and some people try to relate this to changes in habitat, again, the loss of seagrass and the potential shift in where the fish were found, making it harder for dolphins to find their food. The fish kill in 2016 was pretty much a direct flow-on effect of that bloom. There were several cloudy days in a row, and what happens when there are cloudy days and there's that many cells of these phytoplankton in the water is they consume the oxygen. So normally in the daytime, as part of the process of photosynthesis, they produce oxygen. So they raise oxygen concentrations in the water during the daytime, and then at night they and everything else respires, and those oxygen concentrations start to go down. Well, if there's shady days, they don't get as much oxygen produced during the day, and so the dips early in the morning, when it's typically the lowest concentrations, will go below 2 milligrams per liter, which is the level at which you get stress for fish and other aquatic organisms that need that oxygen to breathe. And we did lose about 50% of the seagrasses, about 47,000 acres. So we'll have a look at what that looks like by following this series of maps. The years tick by in the upper right. 2011 is when you'll start to really see something exciting happen. 2007 was the best. 2011, 13, come back in 15, loss again after the 16 bloom. And recently the maps again show that there's been a very little gain, in some areas a decrease in seagrasses still. So the big question was, what exactly happened? Certainly at the time, a lot of people who were working on addressing these issue of, the lagoon and algal blooms and the nutrients that drive that, which typically come from the land, human activities on the land, fertilizers, septic tank leachate, so forth. They had a lot of questions about what was going on. So this is one kind of model, as it were, of how to explain how this tends to happen. The process is called eutrophication, and what that process is an increase in organic matter in a system. So typically it's driven by increased loads of nutrients. You can see the little devil pounding on the system, and that's what loads of nutrients do. But in the system, there are these kinds of shock absorbers that the nutrients don't necessarily immediately lead to a major change in the system. Those nutrients cycle, they're bound to things like sediment particles, and they get pulled out of the system, not available to the phytoplankton or the other plants. There's also, when the phytoplankton do get hold of them, there's typically grazing that helps keep the abundances of phytoplankton down. So in most cases, either the loads get too heavy, as it were, for those shock absorbers, or one of those shock absorbers changes somehow. And then you typically see a system move very rapidly from being kind of that clear water that you can see in the picture of the seagrass with the quadrat on it, to a lot of phytoplankton or macroalgae, in our case phytoplankton. And that picture in the middle there is what's called a Secchi disc that you use to look at water clarity, and typically you're going 0.5 meter to 1 meter or so, and that's about 1/10 or 2/10 of a meter. So very cloudy, as it were, water. Light doesn't penetrate very well, and when that happens, seagrasses don't get enough light, and you get this kind of increased shading, so they can't photosynthesize to keep themselves going. And benthic respiration may increase, and that can drive sulfides in the sediment, which also are toxic to the seagrass. And so you oftentimes get a very rapid loss of seagrass. So we didn't see increased loads of nutrients. In fact, people were working hard to decrease loads of nutrients. So we kind of looked at some of those things that happen in the system. Cycling is one of those things that happens. So this shows you a graph with the shaded area is seagrass carbon. So seagrasses take up nutrients, nitrogen, phosphorus are the main ones, and carbon, and they store them in their tissues. And seagrasses typically turn over fairly slowly, so they're long-lived. They don't cycle very quickly. The 2 red arrows show you places where the blooms led to losses of seagrass, and so we'll track kind of those spots as we go through. Another key part of the cycling are drift algae. So these are larger algae, not single-celled algae, that do not need to be attached. They're quite happy to just roll around on the bottom of the lagoon. They also photosynthesize. They also take up nitrogen, phosphorus, and carbon, turn it into tissue. They're food for some of the inverts and so forth, which then become food for the fish. And drift algae work at a little faster pace than seagrasses, so they're a little more ephemeral. And we saw in the past that they would come on kind of in the spring and early summer, but when the water got too warm, then we wouldn't have as much drift algae. But then at those 2 times where the seagrasses dipped, the shading also affected the drift algae. So you had neither seagrass or drift algae really competing for a lot of these nutrients. And that's when the phytoplankton come in and they churn the fastest. So for a range of reasons, they're small, they're up in the water column and so forth. They capture that nitrogen, phosphorus, and carbon, and they turn it into more phytoplankton. And then you can kind of see how this turns into a bit of a downward spiral. So more phytoplankton, more shading, less seagrass, less drift algae, more nutrients and so forth for the phytoplankton. And that's kind of the concern that people have, is that the system is shifting. It's shifting from a seagrass-based system to a phytoplankton-based system. So we also looked, and there are what are called internal loading sources. In particular, one is what's been called muck, which are sediments, fine sediments with a lot of organic matter, or not a lot, but some more organic matter than the typical sandy sediments in it. And this muck tends to accumulate in places that are deeper. So you look at the map and you can see the path of the intracoastal waterway, which is the dredged channel for boats to go in. And you can also see some patches where there's been dredging to get material for causeways and things and bridges and so forth. And so the muck accumulates in some of these places, and with the muck and the organic matter that's in it, it fluxes, it releases nitrogen and phosphorus into the system. And so it's an internal, what's become called a legacy load, because it's built up over 50 years or more in the system. Grass clippings, leaves, all that can become part of this muck. And one of the questions was, did the system, because of the temperatures and so forth, it's possible that some of the binding of things like phosphorus changed. And so this phosphorus was released, and that drove some of those blooms. Another thing that we know pretty much for certain is that the types of phytoplankton that are blooming in the lagoon have shifted. In the good old days, we used to have diatoms. Pyridinium bahamense there is a dinoflagellate. We had other dinoflagellates. We had some cyanobacteria as well, but they tended to be a little larger than the ones we're seeing now. And so from the super bloom on, we've had things like what are called picocyanobacteria, which are really tiny cyanobacteria. Small chlorophytes, what are called nano phytoplankton. So for nano phytoplankton, you can get about 200 of them across a period at the end of a sentence on a typical page of text. That's how small they are. Oreoumbra is a nanoplankton as well. And then the recent bloom, the one that a satellite image from the 27th of October, the dominant in that one is a different kind of cyanobacteria. It looks like a cyanobium species, but the genetics are being done to figure that out. But all these as we shifted to smaller phytoplankton, they turn over faster. So, they divide more quickly. That's how they reproduce. Some of them have characteristics that make them a little more problematic. Brown tide has a resting stage, which means that it's sitting right there on the bottom. So when conditions are right, you don't need a kind of an inoculum. It's right there already. Some of these new players look like they can do what's called capturing organic nitrogen and phosphorus. So the way it typically works is the nitrogen and phosphorus are in some organism, and when that organism dies, they get released, but they're bound to the carbon. That's what makes them organic forms. But some of these don't wait for bacteria that typically strip the carbon off the nitrogen and phosphorus and release the nitrogen and phosphorus. They can go in and do that themselves, which means they cycle much faster as well. And you don't get kind of a leak of this organic material out of the bloom. Some can maybe fix nitrogen, so they can use nitrogen from the atmosphere. They don't need it to be fixed by the bacteria and made bioavailable. They can get it themselves. Also, partly because they're small and potentially because they have mechanisms to deter grazers. For example, the brown tide produces a mucus coating that can deter some grazers. We probably have less top-down control, less of the phytoplankton are being eaten by small zooplankton, micro or larger zooplankton, mesozooplankton, in fauna, things like clams, or epifauna, things like sponges and so forth that you see on your docks and around on the shoreline. So all this combination means that the blooms, tend to be a little larger, they tend to be a little longer lasting, and they tend to get more intense. So what are folks doing about all this? What's the response been? Well, we continue to track the water quality. So on the left are places where the St. Johns River Water Management District tracks water quality, the blue dots. On the right are places where South Florida does, down in that part of the lagoon. And in fact, some of the monitoring is shifted over to what we call continuous monitoring. So there are these probes that are placed in the lagoon that measure things and record them and also can transmit them, and they're online. So if you go to our district website, you can find data from the sondes that we have in the lagoon, which is on the left. Harbor Branch runs a set of sondes, and they have a website as well where you can find their data. And then another group, the Ocean Research and Conservation Association, ORCA, runs a set that are more focused in the tributaries. So all these data show up. They're typically refreshed sort of on an hourly basis or so, and they're all available online if you want to track it. Probably the biggest thing that's going on is addressing the shared challenge throughout the Indian River Lagoon system. And that's, again, this excess of nitrogen and phosphorus. And this is addressed through the total maximum daily load process, where we estimate what the safe load is, and then take a look at the existing load, find the difference, and then allocate reductions to the various stakeholders to reach that safe load. And there's a margin of safety usually factored in because we know that all these estimates have uncertainty that surrounds them. In addition, the plans to make these changes, to achieve these reductions, the Basin Management Action Plan, or in the case of Mosquito Lagoon, a Reasonable Assurance Plan, all contain an adaptive approach. So they typically have some sort of indicator. For a lot of these plans, seagrass is a key indicator. So how are seagrasses doing in the lagoon? And it's looking at that indicator that says, have we done enough? Not just did we reach the estimated reductions, but are we actually seeing the response that we're after? And so if you're not, then you may go back and increase those allocations so that you do see the response that you desire. So what kinds of things does that translate into? Well, people have kind of taken to saying, well, we're putting the lagoon system on a diet and we're giving it some exercise. So just as you try to lose weight, we're doing the same thing for the lagoon. The diet includes, like in the upper left, that's a stormwater treatment area that the district manages. So stormwater is collected in the canals. We dug a lot of canals to drain the land. That, although water in those canals carries nutrients with it, we take that water, we run it through wetlands and so forth that pull those nutrients out, and then release the water after that. Some of the canals actually went through the ridge that divided the upper St. Johns River Basin from the Indian River Lagoon river basin. And so we're now pushing the water back to where it belongs, as it were. So the upper right-hand photo is a set of pumps that move water out of one of the larger canals. It goes into a stormwater, a large treatment wetland, and then it flows into the St. Johns River, which is where it used to flow. In terms of exercise, it's getting in there and getting out some of these internal legacy loads like muck. And so you, on the lower left, there is a dredge, and that's pulling muck out of the lagoon. The other way to get it exercise is to assist the filter feeding populations, things like oysters, which is what you see on the lower right, because they filter water. And so they'll not only they remove the phytoplankton, which helps keep the water clear, and they store some of the nutrients in their bodies, and they don't turn over as fast as the phytoplankton. So diet and exercise for the lagoon. Other kinds of projects. The map is showing you some of the district projects that have come through cost share and so forth. Putting in advanced septic systems, converting septic systems to sewage, and repairing and renovating lift stations so that you don't have releases of sewage. All those things go on. Certainly the district's far from alone in this. A couple of the major players, Brevard County, they instituted a sales tax that they put towards the lagoon through the Save Our Lagoon project plan. And then the Indian River Lagoon National Estuary Program, and they have their Comprehensive Conservation and Management Plan, the CCMP, and both these groups are trying to address the same issues. So Brevard County will raise about $400 million over 10 years, and the Indian River Lagoon National Estuary Program has about $1.2 to $1.5 million a year that they put into mostly into projects as well. Other counties are also coming up with ways to move forward. St. Lucie, Indian River, Volusia as well. And there's a statewide focus. There's an innovative grants program that the DEP manages, where they're looking at new technology or that might help us with this issue of blooms. Blue, green, and red tide task forces are formed, and those are groups that are meant to think more deeply about how to handle certain kinds of blooms. And then the DEP has their Protecting Florida Together website, partly to gather this information up and make it available to the public as well, so that people can see what's happening and understand what's going on. So just kind of go back and close up with what about the future, which I would subtitled here the question I often get from my executive director: how does all that HIGAD stuff help? What we've learned is that events matter. So most of our management is towards kind of the average, but we've figured out now that we need to take into account events. We may need to leave a little elbow room in the total maximum daily load process for things like that, what led to that 2011 bloom. Nutrients leading to chlorophyll still is a big issue for the lagoon. That is the issue to kind of keep the focus on. But it might be good to understand a little more about the nutrient budget. So not just the load, but what happens to that load when it gets in the lagoon. How does it cycle? We've also learned that the sources of chlorophyll matter, and some of the sources we have now are a little bit more bad actors than we used to have, which again tells us that kind of having information a level below where you want to manage can be helpful. Gives you a base to build on. Also, hopefully conveyed the fact that restoration is underway and planned for the future. So when people say there's nothing going on to help this situation, that's not really accurate. You have various people taxing themselves. You have this IRL Council and the Estuary Program working hard. St. John's District is working hard. The South Florida Water Management District's working hard. All of us trying to get to the same endpoint. But I think patience is probably the key. So it took a while to get here, and it'll take a while to get where we want to go. Thank you very much for your attention.
Thank you so much, Chuck, for that fabulous presentation. If you do have any questions, please enter those in the question box. We had a couple of them come in. One of them was, where are these new species of cyanobacteria coming from?
The chances are good that they've always been there. They just never were dominant. So when phytoplankton bloom, there's a kind of a competition that goes on, and there's a unpredictable, what we call stochastic component to that, as to, you know, who's there at the time that nutrients become available, who gets hold of them first, and then who, you know, sort of takes the race, as it were. We're working for this particular one that's in the bloom that's going on at the moment. That hasn't been reported before. It's pretty nondescript. It's very small. So if there were only a few around, it would kind of go in the other category when we count things, when the phytoplankton get counted. And they're working on the genetics to try and nail it down so that we do understand, you know, who it is. Probably one thing that I should have said is, for the ones that we've had issues with, they're not like the microcystis that you hear about in the southern part of the lagoon coming out of like Lake Okeechobee. So we have no evidence that they have produced toxins, and there's no history, you know, in the literature and so forth that they do. So we're lucky that way, as it were. So.
Yes, so that actually goes into the next question of, is it said to be safe to swim or kayak in the lagoon during these blooms?
Well, I mean, that's kind of outside my realm a little bit, but I think generally speaking, if you see a bloom, it's probably something not to get into. Again, you know, there's no evidence that these blooms have toxins or so forth, but just out of an abundance of caution, I would say wait.
Right.
But I would hesitate to tell someone it's safe and then have them, you know, find that they have some problem.
Yes. So. Absolutely. And this may be another question that you're not the person to go to, but why is the northern IRL not part of the aquatic preserve?
So
not sure.
I think, you know, big chunks are, and there's a lot of conservation land up in the very northern parts. So you got Canaveral Seashore and so forth. And then of course you have this Kennedy Space Center, and you know that's got a whole restricted area there off of that. But the preserve does, you know, move up that direction. So I should go back and say, for the safety part of it, I mean, the DOH and the DEP are working real hard on making sure that people have information about safety. So they come and sample when there's blooms, and they continue to do so until there's no bloom or no toxins, or, you know, and they put up warning signs and so forth. So look for those.
Absolutely. Well, thank you so much. I hope everybody that attended today enjoyed it as much as I did. And I encourage you to register for our next webinar on the cost shares that are going on in our district. A few of those will be highlighted. And if you have friends that want to watch this that weren't able to participate today, it was recorded and will be available on our website. Thank you so much, and I hope you have a great day.